Thank you, Oliver, for the elaborate introduction. Aging research does predate my coming to the NIA, although one might not have that impression from all that was said. And again, I wrote it, so I'm responsible. Thank you, Paul Spiegel and Keith and Elena for the invitation to make this presentation. So I'm going to talk about concepts and perspectives in geroscience, and I have no conflicts of interest because I'm a federal employee. There are hallmarks of aging that have been mentioned, and there are things that contribute to aging, and they can be broken down into multiple ways. I'd like to give you some feeling for how we view it as extraordinarily complex and yet tractable. So there are some functional hallmarks of aging. If you recall from Vera's talk yesterday, you might take home the message that you can beaverize a mouse, but there was another message in there that was, how do you analyze an aging phenotype? And you start with the appearance, you move on to function, and then you would look at lifespan, in pretty much in the order she described. But, and there are historical reasons to look at lifespan, and there are practical reasons to put it as the last. But one of the things that we are now trying to promote, specifically my division director, Felipe Sierra, is the concept of resilience as a metric and a hallmark of aging. That is, resilience declines with age. But there are other features on here, some of which are mobility, strength, and weight comprise the key components of a frailty index as promulgated by Linda Fried, now up at Columbia. But there are other features that where you can analyze function, and you can do it in a range of organisms. It may be more challenging in some organisms than others. And then there are some clinical hallmarks of aging. There's been a lot said about diseases that are associated with advanced age. Some of them are lethal. Those are underlined, cancers, heart disease, and so on. Some of them are syndromes for clinical purposes, such as frailty, which is here, which Linda uses to tell you if a person of a certain age is likely to survive a surgery of a certain severity. But there are things that decline as well: hearing, vision, loss of mobility, etc. These are not inherently lethal losses of function or declines. They are disabilities if they become severe enough. You can look at the hallmarks of aging from the point of view of aging organs. You can look at them individually. Certainly lung function will decline. Muscle will decline from its peak, which would be for a male at around the age of 32. Bone, which has a very strong sex-specific change with age. Fat depots, etc., the heart. And these are interconnected. So at this point, let me say that this Piet Mondrian-like interaction here is to, or set of lines, is to suggest strongly that there are interactions between the various components. We know that the kidney interacts strongly with the heart. We know that the heart, brain, and fat depots interact, along with some bone-brain interactions and muscle-lung interactions. These are all being established as parameters of aging. So you can look at them individually, and you can look at them as they interact with each other. And of course, there are these cellular hallmarks of aging. Cell death was mentioned yesterday by Pam Maher. There are a lot of regulated forms of cell death, ferroptosis being one of the more recently labeled or relabeled ones. Of course, cell fates change with age, something that I will discuss, but there's a stem progenitor relationship, and from progenitors they can give you different cell fates. There's been over some time a little bit of uncertainty as to which cells were the precursors for which fates, especially fibroblasts and fibrosis, occur during aging, but that's being settled now. And of course, senescence is well known. Immune senescence, also mentioned yesterday by Andre in his talk, and they all interact. There's cell non-autonomous aging. My institute is funding now, in response to a request for applications, some work on cell non-autonomous aging. And of course, the senescence-associated secretory phenotype is quite famous from the work of Judy Campisi, who unfortunately did not speak yesterday. But all of these things will interact. Geronik factors is a neologism of my creation. It's debatable as to what it means for most people, but for me it is something that applies to a specific situation that Justin Rebo mentioned yesterday, which is heterochronic parabiosis or heterochronic blood exchange. I'll elaborate a bit more on that later. And then there are the molecular hallmarks of aging. There seem to be 9 on this. If you get tired of drawing these lines, that will limit the number that you come up with. But they can be arranged in any order. Damage features quite prominently in the biology of aging, and it can be overlaid onto any of these other hallmarks. Changes in mitochondrial function, mitochondrial quality control. We know that various aspects of metabolism interact with other features. Not everything gets the same play. You don't hear a lot about cytoskeleton in terms of aging. But here's a view of the complexity of the problem. You have cellular and molecular features that are the basic biology of aging, and then up here you have the more clinical aspects, which are functions, the morbidities, multimorbidity being an index of aging, at least it is an index of aging. So that's one way of looking at it, that these all interact, and that this is, from a research perspective, a foundation, but this is what you're really trying to affect, especially in the development of anti-aging therapies. But it's a little bit more complicated in that you have medications that people take, quite a few, diets, which change frequently. Not everybody eats a pound of carrots a day. There are various behaviors that we engage in, some of them fun and bad for us, and some of them miserable and good for us. And then there's the environment, of course. So in trying to go from the pressing questions of human aging, to how you examine these in the laboratory through the proxies that we generally support, which are everything from yeast, which is a fungus, to non-human primates, you have to take into account how you understand the different morbidities, functions, the interactions of the organs. Dr. Suleiman yesterday made a point about the ulna bone, which is quite well taken. It can be an organ that you think has a specific function. So in the fly, which doesn't have a heart and doesn't have a closed circulatory system, you're looking at the corpora cardiaca, which also has some secretory functions that you don't find in the human heart. And yet you can learn a lot about cardiac aging by examining the fly. You just have to be cautious in being and circumspect in how you interpret the results. So it's true that the biology of aging is complex. It's true that in terms of hallmarks of aging, it's complex. But about 25, 30 years ago, the NIA, primarily under the impetus of Dr. Anna McCormick, since retired, set up a series of requests for applications. She managed to do it in 3 iterations on longevity assurance genes. And what put biology of aging on the map was lifespan. And lifespan is tractable because it's binary. Primarily, things are alive or dead, and you can usually tell the difference. Well, so there are circumstances where you can't. For example, you might judge that a worm that doesn't respond to being poked with an eyelash is dead, and that worm sits on the plate, and you know where it was, and you come back a day later, and it's moving. So they do become quiescent for a while and unresponsive. But it's usually not enough for them to be statistically significant. And sociologically, there are issues, and legally, there are issues of what constitutes death. So in the biology of aging, we've simplified it somewhat by asking about lifespan, and we've asked for in terms of what are the genes that affect lifespan in a laboratory animal, keeping in mind, which we did not and still do not, that these have undergone artificial selection. So as Rich Miller so eloquently puts it, these are mouse-like objects that are being studied in the laboratory. They're not the mice that you would find in the field, although they share some characteristics. You can put a running wheel in the field, and at night, the field mice will run on it. And there are pictures, movies of that from Steve Alstead. So what you have is a control population or a reference population in the laboratory, and you are looking for alleles of genes that will increase lifespan. The famous one is the DAF2 mutant in flies, which is an insulin receptor mutation. There's a raft of them. In humans, most insulin receptor mutations from the work of Simeon Taylor result in a diabetic condition or are linked to diabetes. It's not the only cause, of course, not even the major cause, but it tends to be deleterious. But in lower organisms, you can interfere with the insulin signaling, down modulating as opposed to obliterating it, and you can get an extension of lifespan. So there are also a raft of single mutations that have been made in mice and other organisms that shorten lifespan, but the sine qua non for what is a gene linked to lifespan is, does it increase lifespan? There are a lot of single gene mutations, and then there are also mice such as the SAMP8 that Pam Mara mentioned that's not a targeted mutation, but they affect lifespan. So this established it as a discipline. Now there are more than 400 genes that have been linked to lifespan. It's fortunate that not all 25,000 of them were, because that would just tell us that we barked up the wrong tree. So does every animal benefit from whatever the intervention is? So it could be a genetic intervention, it could be a pharmaceutical intervention, the second being of more interest to this group. But everything dies in this experiment. So they will just die at different times, and we used to be concerned with what is the cause of death, because again, that was an easy question to answer, but it turns out not to be. We support a program in what's called geropathology, which takes the corpses and examines them for what was the burden of disease at the time of death. That's an interesting problem, but what it really tells us is that perhaps lifespan doesn't translate that well because the causes of death are endemic to a lifespan study. Maybe you need to think of it in somewhat different ways. That doesn't mean abandon it. It just means what can we add to it? And what we can add to it is thinking about the health of an organism. So in the biology of aging, from this diagram, there is another property besides imitating a famous artist, and that is it shows you the properties of a biological system. This is a representation, maybe not topologically accurate, but it's a representation of a network. Nodes that are linked to each other in some fashion. And that suggests that if you were to plot in a population a conglomerate of hallmarks of aging as an aggregate score, you could view that as being similar to a genetic landscape. So evolution has populations, and the populations are subjected to changes in the environment. So it's not just large allele A, small allele A that's being selected, it's the entire genotype that's being selected. So across here, there are a range of genotypes, and this could be fitness. And as the environment changes, some of these become more fit in response to, are more fit in that changed environment. These will survive, and these will eventually be eliminated, and that's how you get speciation in a nutshell. So one of the ways you get it. So in terms of aging, so this is not an evolutionary question, but it has some similarities. You have a population, and you have younger and older phenotypes at any age. And if you look at the work of Dan Belsky on the Dunedin study, you'll see exactly this kind of analysis is presented. There are people who are all 38, and some of them seem to be younger, and some of them seem to be older. And to my knowledge, there is no evidence that in that study, the younger are all eating carrots. So Vadim Gladyshev mentioned yesterday that you can look at signatures of aging. And I think this is a genius thing to do. And I think that because we've tried to discuss this within my institute and gotten no traction, so it always looks better when somebody from the outside does it. But it's also genius because it is. So I have no idea what Vadim's signature looks like, but I just made one up. So you can recognize some parts of it. That could be a V. There's no way to know what that is, but that could be an M. But this looks like that, but it's not the same set of letters. That might be a G, or could be a J, I don't know. But the thing is, if you if Vadim were to have a signature or something, whatever his signature is, and he presents it to the bank, and that's his official signature for the checks that he would sign, which of course is archaic technology, but still it makes a point, the bank would recognize this as that. So you don't necessarily know what the components are or what their relationship is in terms of causality of aging or permissive of conditions of aging, but you can recognize it. And I believe one of his points was that you can use signatures for phenotypic screening. Not the only one to make that point yesterday, but phenotypic screening for compounds that affect aging have two valuable purposes. One is their potential anti-aging drugs, and the second is they are useful for research into the biology of aging. So the signatures of biology actually are done as composite indices if you look at some major benchmarks of biological processes. So fetal development has a Ballard score associated with it, and you can do a whole bunch of different metrics and tell where in the course of development is this fetus. And you can do this for any mammal, and there are similar indices for Drosophila and other invertebrates. But importantly, there's at birth, you can ask, how healthy is the baby? That's a question that we are asking of ourselves, how healthy are we when we age? So here's at the other end of at least breathing life, there's an APGAR score, which was created by a pediatrician at Columbia University named Virginia Apgar. This was a reverse acronym, so I can't remember what the individual letters stand for, but R is respiration, but it's a scale from 0 to 9. And where you are on that scale determines your relative health, and then which component of it scores low is the thing that the physician needs to pay most attention to. For maturation, which is the transition from birth through reproductive maturity, there's a Tanner scale, which gives you all these indices of primary and secondary sexual characteristics, and you get a composite score, and you can see where a person is in terms of progression. Are they ahead of the curve, behind the curve? Do you just leave them alone? They're teenagers, you should, or do you have to do some kind of intervention? And then there's aging. Well, we don't really have a composite score at this point, but the signatures might be one. So Felipe Sierra has been promoting resilience. Now, there's no Sierra Resilience Index, and I'm not going to get a raise because I put his name on it, but you're going to have healthy at 0, or there will be a range. It probably should be N to 0, but what this is doing is predicting an adaptation to stress. So we are looking a lot at biomarkers from what's in your blood or some other accessible tissue or material, and we're basically taking a snapshot, and we're asking how does that compare, and we can take that snapshot linearly, as Luskarten does, and you can track what's going on, and you can make inferences from that are correlative. But there are all sorts of things that you can do in a laboratory animal or even in a home-housed pet that could constitute resilience. So one of them is returning to resting heart rate. You exercise to whatever extent is possible for that person or animal at that age, and you ask how rapidly does the heart rate return to its resting level. Now, George Taffet has shown that this is not monotonic, that heart rates can return too slowly, that's bad, or too quickly, that's bad. Each indicates a particular pathology that can be treated. So it's a good diagnostic. There's recovery of muscle function after inactivity. This is certainly important for anybody who's bedridden at an older age, and how much can you recover. It's also important in studies, which I will talk about later, if I don't run out of time, that have to do with, you need to do more than just look at cells and molecules, you also need to look at function. There's adaptation to acute stress, and there's distraction. If you happen to remember Keith's talk yesterday, no, I'm sorry, not Keith's, Grinberg's talk yesterday. He flashed an image of a young woman in a store trying to make decisions about what to buy, what food to buy, and all of the components that you can break that down in an engineering point of view. In the background of that slide was a guy shopping, and he had his cell phone to his ear. So if you're going to measure distraction, you're going to have to take, in a human population, you're going to have to take into account your generational reference point. I would not shop with a cell phone next to my ear because I couldn't read the labels. I'd be distracted. Whereas this young man can't shop without a cell phone next to his ear because that's how he's accustomed to doing things. The same is true for laboratory animals. If you have a mouse in a cage with nothing to do versus a mouse in a cage with toilet roll paper or some distraction, some activity, the distractibility of that mouse is probably going to be different, and therefore their neurological functions that you're trying to report on are going to be different. So if you go to perturb and look at recovery, you might get a different outcome. But basically, this is what we're all trying to do. We're all trying to get interventions against aging. Whatever it is that we mean by aging or how we classify aging, what we'd like to do is have people healthier at any age. That's the NIH mission, and we'd like to have people healthier at older ages. That's the mission of the National Institute on Aging. So what you have is a starting population that has a distribution, and you do an intervention over time, and you have a reference population that may have been at this point, but at a different age, so it's not a longitudinal study for the reference population, but your ending population is longitudinal, and you can sample along the way. And what you're trying to do, really, is shift the distribution from there's this part of the population that's older, now you have this part of the population that has the younger aggregate score, signature, whatever you want to call it. It's unlikely that you'll have one thing that will affect everybody in a positive way, so you'd like to have several things that would affect people individually in a positive way. And if you collect samples along the way, as has been done in many longitudinal studies, some of which are supported by my institute, you can attempt to find what here will predict this shift, and what here will predict the failure to shift, or what here will have shifted from young to old, which would be an unintended consequence. So, my evil twin yesterday talked about this. What does the FDA need for interventions against aging? So let me start with, we are trying to develop a request for information. So I can talk about that. I can't talk about what we plan to fund, but I can talk about what we plan to ask you to tell us. You are the people who are working in this space. We are going to request information on several areas that have emerged from our discussions with the FDA. We don't know that they're going to agree to it. In our discussions, we've asked them, if somebody's going to set up a clinical study on an anti-aging drug, what do they need to do? What do they need to have in terms of the parameters that go in and the parameters that come out? Really? 1 minute? Okay. So you can read what you have up here on the board. You have a lot of biomarkers that are defined by the FDA that come in various formats and various forms. So in this case, signatures won't be enough. You'll have to have you'll have to know what these various things are when you go to set up a clinical study. How much time do I have? 5 minutes? Okay. Quickly then, for the next 35 slides. Slowing the rate of aging, this is the Geroscience hypothesis as articulated by Steve Austad and James Kirkland. So the question actually is, can aging be altered? Does old biology have the capacity to respond to a young stimulus, or can it be made to have that capacity? And on the flip side, can we learn something about how aging is accelerated? Because there are lots of things where aging is accelerated. So what we support are laboratory animals, which are proxies for lifespan extension, regeneration, and rejuvenation, and they are, in certain circumstances, models for diseases of aging. A lot of what we know actually comes from a very unusual set of experiments pioneered by Irina Conboy, and which Rebo mentioned yesterday, and this is his paradigm. Heterochronic parabiosis is you stitch a young and an old mouse together. The young mouse in some cases gets older, and the old mouse in some cases gets younger. And in heterochronic blood exchange, you just move the blood from one to the other, and you look for the transposition of the aging phenotype. You want to discover the factors that are involved. Geronik factors is a term that are used to describe what it is that's responsible for this unusual biology to take place. You have more options here as to how you can perturb it. Exercise improves health. So you can exercise, you can change the diet, you can expose them to chemicals, you can take them early in the day, late in the day. There are a lot of parameters that you can change here that you cannot change in this experimental paradigm. GDF11 was asked yesterday in a question, This is one of the things that got the field going in terms of its ability to rejuvenate some old phenotypes, but I would like to point out it's an extraordinarily complex signaling system. Here's GDF11. There are multiple binding partners. There are alternative ligands for the receptor and the downstream signaling that's involved in aging. So we've supported a number of these things. Fabrizio Ambrosio works on Klotho in muscle regeneration. This is a paradigm where you injure the old muscle. Justin showed slides of this. You injure the old muscle, there's fibrosis. If it's parabiosed to a young mouse or given young blood, there's less fibrosis. Why is that? Ultimately, it's a competition between fibrogenic, adipogenic precursors, which have a cell fate decision to make. They can be fibroblasts, or the muscle stem cells can become myoblasts. As in the signaling by GDF11, cellularly, this is an extraordinarily complex set of circumstances. Some things may be functioning pharmacologically. That is to say, perhaps the level doesn't change with age, but if you add that thing to the organism, you get an improved outcome. So that's a candidate pharmacological protein or polypeptide. Klotho also appears to be anti-Geronik, but from the work of Ambrosio published, I don't have the reference here, but it's on another slide. Actually, the muscle stem cells make Klotho, and that expression declines with age. So you're getting another pharmacological effect by giving Klotho extracellularly. It turns out that the Klotho is moving in extracellular vesicles. The coding for that moves in extracellular vesicle. It's taken up and it has a response. Tom Rando is showing that C1q, part of the complement pathway secreted by macrophages, has this effect on cell fate decision. So there are other cellular effectors. Macrophages can go from a young mouse to an old mouse in a bone repair model and affect the precursor. So this is a young macrophage affecting an old precursor cell to affect the osteoblast versus fibroblast fate decision. The important point of this is the old progenitor can respond if it had the appropriate signal. Here's one where you inject young bone marrow cells into an old bone marrow, and you get rejuvenation in the brain, going at a distance. You measure activity, and the activity is increased by this relative to the old. Here's a more complicated figure that I won't dwell on, but it's basically things don't cross the blood-brain barrier. In old animals, the microglia do not function well, and this is because of a leukocyte transmitting some signal across the blood-brain barrier that can be blocked by blocking the docking protein. Sometimes the macrophages move from one to another, and in this case, they affect remyelination. In this case, it's the macrophage secreting activin, which is part of this complex signaling pathway. I can't go into that, unfortunately, but except to say that there are ways of doing these experiments with genetic modification of the mice that you can ferret out which genes are affected by the candidate Geronik factor. In this case, the work from Heinrich Jasper's lab. Here's one that was mentioned briefly in a panel from Keith's talk, ENAMT. This is an enzyme that's in circulation. Yoshida et al. Claim that it is a predictor of remaining lifespan, something that an ethicist might be wondering if that's a good idea to predict. There's a male-female difference. In mouse and humans, they both decline with age. Kladyshev mentioned that you can compare young and old. This is a comparison of young and old. It's not a parabiosis. This also is an enzyme that seems to be moving through an extracellular vesicle. So aging can be altered, it can be decelerated, it can be accelerated, and what's doing that? You name it, and it will do it. That is, you can find it. Proteins and polypeptides are where we started as ligands. It's not enough that a ligand just changes between young and old. You have to know where you are on the saturation curve for its binding, because if it's already at saturation, changing it twofold won't matter. Lipids were mentioned yesterday by Mar. Enzymes in the ENAMT, extracellular vesicles also there, and cells. Cells are particularly interesting because you can when you induce pluripotency in a cell and then differentiate it, that's largely speaking a younger cell. So I was going to illustrate cardiomyopathy as a genotype hallmark of aging interaction, but I'll just jump to the chase. But I will point out that the next speaker created the mice that were important in this experiment. It's basically you take the genotype and you shorten the telomeres progressively in the mice, and what happens is that you get the cardiomyopathy in the mouse. So in the Duchenne muscular dystrophy, you make the null, you get the muscle, skeletal muscle phenotype, but not the cardiac phenotype. And if you shorten the telomeres, you get the cardiac phenotype. So it's we need to see more of this kind of thing, of where you're looking at the hallmarks of aging interaction with a gene causing allele. There are a lot of opportunities there to do things. Helen Blau's work there on the Duchenne's and also on cardiac myopathy, she's presenting evidence that for a lot of these herited cardiomyopathies, not fully penetrant but partially penetrant, shorter telomeres are important. So to some, or eyes, in geroscience, there are hallmarks of aging. They may be permissive of diseases by interaction with genes, et cetera, or environmental conditions. They may be causal. We need them to be diagnostic. Composites are recommended. And what's in your body? Does it make you age, and can it rejuvenate, are the two questions that you, through your work, will answer. Thank you.